The last thing I want to tell you about in this class is probably the coolest. And that is that macromolecules carry information. And they carry information because they have got different ends and because they have got direction. And if you think about it, all languages have got some kind of start and stop signals to them, and there is some direction associated with them. And the language of life is exactly the same. So let us write down that macromolecules may have order and polarity, where polarity would give some kind of direction. And these two attributes together comprise information that can be used by the cell. The two classes of macromolecules that do this par excellence are the nucleic acids and the proteins. And so we're going to talk about both of those, nucleic acids and proteins. The nucleic acids, as I said, carry the information of hereditary. The proteins carry the information of just about everything else. So these two classes are what we'll discuss over the next few minutes. In order to do that, we need to come back and consider the nucleic acid polymer. The nucleic acid polymer has two different ends. It has got a so-called 5' and a so-called 3' end. And you can see that if I draw you a very rough schematic of how a nucleic acid polymer is put together. I'm going to write 5', and next to it I'm going to put P for phosphate. The phosphate is covalently bonded to a sugar, abbreviated S, which is covalently bonded to another phosphate, another sugar, another phosphate, another sugar in my schematic. And then I'm going to write 3'. And sticking off from the sugar, I'm going to write the bases, base 1, base 2, and base 3. OK, so let's look at what I've drawn here. PS refers to a covalently joined sugar-phosphate backbone. It's the thing that holds the nucleic acid polymer together. The bonds that join the sugar and the phosphate have a special name. They're called phosphodiester bonds. And they join the phosphate to one of the hydroxyl groups on the sugar. Let's think now, going back to that polymer. You can see I've written 5' and 3'. At one end of the polymer, this has to do with polarity. At one end of the polymer, there's a 5' end, and the other end, there's a 3' end. Those are two different ends. They're chemically different, and the cell can distinguish them from one another, and that gives you part of the information coding in nucleic acids. So the 5' end is the phosphate that's joined to the 5' carbon on the sugar. And the 3' end is the 3' hydroxyl group that, again, is on the sugar. The second thing that you need to know is that this polarity is superimposed with the bases and the sequence of the bases along the polymer. So base 1 is near to this 5' end, then base 2 and base 3. So there is base order along the polymer. And then the last thing that you should know is that a new base will add on to this 3' end. So this base 3 was the last one that was added to the polymer. And when a new one comes along to extend the polymer, it will add to that 3' end. So the 3' is the last base added, and the next base adds to the 3' hydroxyl group on the sugar. Okay, let's take a look at a slide to help us there. This is a fragment of a DNA polymer, and there is the 5' phosphate and the 3' hydroxyl circled in blue on either ends of the polymer. And I've also circled this complex phosphodiester bond that holds the sugar and the phosphate together. And the bases are kind of separate. They're hanging off the sugar-phosphate backbone. If we look at a schematic of this, I've drawn you the sugar-phosphate backbone, the base order, the first nucleotide, the last nucleotide, the free phosphate group at the 5' end, and the free hydroxyl group on the last nucleotide. The direction of polymerization is from 5' to 3' because you always add onto the 3' end. But when we write the sequence of the bases on nucleic acid, we don't write the sugar-phosphate backbone, we just take that as a given. What's really important are the base order, B1 to B6 in my diagram, or I've put a real example there, and the 5' and the 3' ends, and you always, always, always write those 5' and those 3' ends. Doesn't matter if you've been working with nucleic acids for 30 years, you always have to write 5', 3', and the base order between. There is no exception ever, ever, ever. Okay? It's one of the most important things I can tell you. Okay. The order of the bases relative to this 5' and 3' is the information in nucleic acid. It's what the genes are. It makes you who you are because of this, these two attributes of nucleic acid. The polarity, the 5' and the 3' end, shows the first to last nucleotide added and the direction in which to read the information.